All right. So there's a picture of what the 11th edition looks like. There's the 12th edition. And you'll notice what I've done on the side of the board there. Chapter 1, and then I've got a Roman numeral outline, 3 main sections. Those correspond exactly to the subheadings in Chapter 1 from your textbook. And I'll do that for you every time. I should be able to get here in like 5 minutes to put them up. This I do for 2 reasons. One, as I'm lecturing, I'll use this. It's the same as in your textbook, so on your notes, you'll actually have what amounts to is an outline of each chapter. That will be really useful tonight, hopefully, or maybe this weekend when you're reading the chapters. You'll have an outline of, hey, this is what the book's saying, hey, this is what we talked about in lecture. Second, if you get lost, maybe you're nod off, maybe you're thinking about something else, or maybe you have just no idea where I'm going, oftentimes we can come back here and you're like, James, where are you? I'm like, ooh, we're right here. And then you can at least start your notes again, boom, and it will make sense. So that's why I do it, and it corresponds exactly to what you find in the textbook chapter. All right, physiology. We need to know what it is. We need a definition. What is physiology? Yeah, study of body functions. What do you guys do in your anatomy class? You studied the structures, the what, but you also did more than just what they are and where they are and where they're located, right? You did what's called functional anatomy, what it is, what it does. So you talked about what the heart does. You talked about what different parts of the brain do. You talked about what arteries do, right? So you did some physiology already. We're going to add to that. So we definitely do the what. We have to know what the what is, and then we add to it, well, what does it do? But then we're going to go into much more detail. Let me give you just so you may have talked about what the body does or what a particular organ does, but you probably didn't talk about what it does under different circumstances. So I can make this statement: body function is not static. Physiology would be a piece of cake if we were all just sleeping like all the time, right? We were always in a prone position. Our heart rate was always really, really low. There was no stimuli. We were just like vegetables, right? I mean, it would be simple. But right now, you guys are sitting down. If you stand up, your body has to make adjustments to its function. The heart has to generate more force. Your vessels might constrict in order to deal with now we're sending more blood to our legs as we're moving them. Temperature, another great example. It was pretty freaking cold as I made you wait outside the lab this morning. You had to make adjustments to your body temperature so that it wouldn't go hypothermic. And here, I'm actually feeling a little warm. It's warmer. Do you think your bodies have adjusted? Yeah, you have adjusted your function. So one of the things that makes physiology hard is not just sort of describing like what the body does, but you have to describe what the body does under different circumstances, and that means what the body does to change its function based on the circumstances. That starts to get complicated because there's lots of different circumstances. We're obviously not going to do all the circumstances, but we'll cover a fair number of them. That makes it complicated. So we could say something like the body alters its function. And I'm just going to say to meet demands. Any examples? I do some examples. We'll do blood flow. Blood flow is a good one. You start moving around, your skeletal muscles are going to be using energy and using oxygen, producing carbon dioxide. Your body has to make adjustments so that those muscles have adequate oxygen, that the CO2 that they produced is gotten rid of, that the nutrients that they need is supplied to them. So that's an example of how the body alters its function to meet demands. Skeletal muscle activity, you have to adjust blood flow to deal with it. What happens to your blood sugar after you eat? Yeah, it goes up. You have to make adjustments to deal with that excess blood sugar. If you haven't eaten food in a while, like if you guys didn't eat breakfast this morning and you skip lunch, like right now, what do you think's going to be happening to your blood sugar? It's going to be hopefully being maintained, but it's not going to be high, it's going to be low, and you're going to have to make adjustments to help keep it at least some sort of normal, possibly slightly low level, so that your brain can function. So just as another example, we can say something like blood glucose with, how about eating and fasting? Regulation of reproduction. At some point that you can remember, you didn't have a functional reproductive system, right? You were little boys, little girls. Wasn't much going on in the external genitalia, ovary, testosterone, testes, ovarian hormone situation. But at some point, you started to change, and that these changes are now happening in a different time frame or different time scale in your life. It has to be regulated. We don't pop out of the womb ready to procreate. We gotta wait 13, 14, maybe years, although it's earlier and earlier for some people. That has to be regulated. So there's just another example of something that's regulated. I want you to look at the time scale on this. How fast can you change blood flow to muscles? Like right now, if you jumped up and started running, is that going to be fast? Yeah. How about blood glucose levels when you start eating a meal or after you gorged, now you're like digesting? A little bit slower. How about regulation of reproduction? Maybe female cycles. That happens on a monthly scale. So these are not just changes that have to be monitored and regulated. We have time scales. Really fast, minutes, tens of minutes, hours, or even scales that are in terms of months, or if you talk about onset of reproduction, or it's waning of reproduction for females at least years of their life. So not only are we going to have to deal with changes and meeting different demands, sometimes those demands are fast, not so fast, or really long-term and really slow. That makes it complicated, because Jesus, it would be easy if we only had to worry about one time frame. We've got massive frames of time to deal with. So that's another thing that makes it more interesting or more complicated. One more. So I've been talking about changing to meet demands. And what we mean is essentially that if you change body functions to meet demands, we say that body functions have to be regulated. If you need to increase blood flow to skeletal muscles, you need to know when. You need to know how much. If you want to adjust blood glucose levels, you need to know what they should be, what they currently are, and how do you get from where they currently are to where they should be. That's regulation. Regulation is complex. It often involves multiple organ systems, multiple effectors. It's probably the most difficult component of physiology, keeping track of how do you regulate or how do you control these adjustments to meet body function demands. And unfortunately, in order to do regulation, we really have to rely on regulatory mechanisms rely often on molecules. Not always. Sometimes we can get around it. But most of the time when we're talking about regulation, we're talking about regulatory molecules. Hormones, ions, neural impulses are basically the movement of ions or the movement of molecules. How many people think in terms of molecules? Like you can't see them, right? They're like more than microscopic. They're like nanoscopic. You can't see molecules. You can't touch or feel individual molecules. In anatomy, life was easy. You could see it through the microscope, or you could like feel it like a cadaver. It was like a bone that you wave around. But you can't wave around a sodium ion. You can't pick up a glucose molecule and be like, ah, yeah, it's glucose. I know you. You're sugar. You have to imagine what it might look like, and we have chemical structures that can do that. But I end up writing things on the board like this. Na plus. That's a sodium ion. We've never seen a sodium ion, and I don't know if we probably could see them through an electron microscope. But we won't have an electron microscope. In this class, we will never see sodium ions, but I'm going to talk about them as if they were our best friends. So you have to be able to sort of adjust your brain and be able to talk about abstract concepts, like things you cannot see. You could maybe say you just don't believe it exists, and that's fine. All the evidence says it exists. We'll be talking about it as if you could see it. One thing that I find is helpful is if you actually imagine that you could see a sodium ion. You can imagine a little spherical ball floating around in the water, and it has a positive charge on it. And if you can imagine that, you'll probably be fine. Think you can imagine that? Maybe. Or how about a glucose molecule? You're eating some food. I guess I put sugar in my coffee. I put sucrose. But I can imagine that there's little sugar molecules like floating around. They're microscopic. But if I could get them into my blood, then I can break them down, and my cells can use them for energy. Can't see them, but I have to like imagine and feel comfortable. Yes, I know they're in there. Not easy to do because it's something you can't see. But that's what we're going to be doing, and we do a lot of it. There's one more thing that's difficult here that I'm not going to write, I'm just going to say, and it's basically physics. There are some cases where we'll actually be talking about the physics of things. Flow of blood, movement of gases across the membrane based on partial pressures. You guys remember partial pressures from chemistry?
Yes.
Good, someone does. We'll refresh it when you get there. It wasn't useful probably then, except for to solve problems. We'll actually see that it is useful for predicting how gas is going to flow and why you can breathe at certain altitudes and why you can't at others. So we'll see that it's useful. But there is some physics involved. Hopefully your chemistry instructor did a good job of giving me that physics, so it won't be like hearing it for the first time. All right. That's physiology. That's a lot. That's complicated. That's what makes it hard. It's never as simple as this is this does this, done, next subject. It's like this is what it does. Other times it does this, other times it does this. How's our brain regulated? How's our endocrine system regulated? What happens when things go wrong? These are the molecules involved. All right, now next subject, and we'll do it all again. 2 areas of physiology that I will mention from time to time. We call these maybe specializations. Pathophysiology and exercise physiology. What's pathophysiology? Yeah, it's the study of disease, so the pathologist presumably determines what disease killed a person, although mostly they just do that section of looking at tissue specimens. But yeah, it's the study of disease. One of the reasons that we're studying physiology is we want to be able to help people when their physiology begins to fail, when things start going wrong. There are lots of really good examples of the types of diseases or conditions that people get that highlight the normal function. So for example, if you look at type 2 diabetes, and we know that blood sugar is high during type 2 diabetes, people are now able to produce insulin, or their insulin that they do produce doesn't work. That gives us a tool to remind us, hey, well, what's insulin good for? Insulin's good for bringing down blood sugar. Insulin is a hormone that's good for regulating blood sugar. So I'll talk about diabetes. Diabetes is also good because a huge proportion of Americans are going to get diabetes. If you keep eating sugar and do not exercise, you can pretty much guarantee you're going to get diabetes. So that means that if you guys don't get it, your patients certainly will have it, a good percentage of them. That makes it really useful for us to talk about because I don't know if you guys have ever noticed this when you go to the doctor, but like when they explain stuff to you, it takes like, I don't know, 3 minutes and then they give you a script and then send you out the door. Most patients that you're going to deal with have no idea what's wrong with them in a concrete way because the doctor has not explained it. Because the doctor
It's usually on
has
the
like,
paper.
well, yeah, but can you read it? Like, just imagine like an 80-year-old lady who's got heart failure. She can't even understand it. I know this from personal experience. My mother-in-law has no idea why she shouldn't be doing these things. The doctor does not explain things to her. It's the nurses, it's the other medical professionals who are going to be in a position to spend more than 5 minutes with a patient and can help them understand what's going on. That is actually of value, which is not going to show up on paper. In addition, if you know what someone's medical condition is, you understand what is going on, you'll be in a better position to really understand how to help them and why you really need to do these things. They're not just abstract things, give them this, whatever, doctor says so, I don't care. You're going to be like, yeah, if I don't give them this, they're going to start accumulating fluid. If they start accumulating fluid, their blood pressure is going to go up. The blood pressure goes up, they're going to lose their rapid. We're going to end up in kidney failure. So you'll actually have a really concrete understanding of the underlying causes of disease, how the medications are supposed to help. Probably makes things a little more interesting than just filling out a chart doing what you're supposed to. Exercise physiology. How many people here consider themselves athletes? Jocks? How many people don't like jocks or athletes, or just distaste for athleticism in general? I have a distaste for athleticism in general, mostly because I've never been good at any sport, ever. But exercise physiology is not about being good at athletics. Exercise physiology is looking at how does the body alter its function to meet one of the most demanding challenges you can subject yourself to. Exercise. Cardiovascular exercise, moving your muscles, is really the biggest challenge that your heart will ever face. So it's useful for us to use it because it's a challenge, and it's a challenge that everyone has to meet. And even if you don't like exercising, I know you like walking to a car, to get a snack. You appreciate exercise when you lose your mobility and your heart starts to fail and you actually walk 3 steps and you're out of breath. Then all of a sudden you're like, geez, exercise physiology would be nice about now. So it's not about sports. It's not about athleticism. It's about, geez, when you move your muscles, this is a whole new sort of set of demands that your body has to deal with. How does it meet those that allow you to move around in order to be functional? So we talked a little about exercise physiology. It's also really easy to manipulate. I can make you guys run around the building once. We've just changed your variables, or at least threatened to change your variables. So it's something we can use in class to demonstrate things, unlike pathophysiology where we don't have lab rats that we make sick or give them cancers or anything like that. We can test on. Questions about physiology? Yeah, Terrence.
So the definition of exercise is?
Just how about function during exercise? Yeah, and really it's the changes in your body that allow you to exercise. Good. The next section in your textbook, I just wrote up there as capital letter A, scientific method. I want you to read this section. It's important. I just don't have time to go over it in detail. Something about read. Let me translate read. Read means if you read it, I won't test you on it. That's what it means. All right. So I'm going to have confidence that if I write read, that you'll just read through it. Don't memorize it. It's not about memorizing all the details. It's just about reading it and being like, oh, okay, cool, I can handle that. That's what it's about. So you promise to read it, and then I won't test you on it. Everyone must promise. All right, good, excellent. I like it. The hand is up. In the simplest terms, everything in your textbook should be there because of some scientific experiment or set of experiments that has validated an idea. There are going to certainly be things in here that are not quite correct. That is, there's an idea or a concept that might not be true. It might be almost true, but it might actually be false. That's fine. Science doesn't care about that. Science is all about getting something that works. If it is false, we hope that somebody else will come along and make it right. But at this current moment, I guarantee there's things in your book that are not quite right. Or maybe it's not the full story. It's like half a story. The key is it useful? Does it allow us to make good predictions? Does it allow us to understand things that are happening in a meaningful way? And if it does, it's useful. If it doesn't, we chuck it out and try and figure out what's really going on. So read it. It's not gospel. It's science. It's based on scientific experiment. We assume that the scientists did a good job and it's accurate, but we're really just taking their word for it. That lets me get to homeostatic feedback. So here's the main concept for the entire course. Just about every single lecture there is something involving homeostatic feedback. There's a little blurb there about clever in art history. I'll just read that section, and really we're going straight to homeostasis. Have we heard this term before? Homeostasis? What does it mean? Balance, that's good. Equilibrium, that's pretty good.
Maintaining the internal environment.
That sounds like something on the textbook, which is awesome. Maintaining the internal environment. That's good. Let's some better chalk. Nubs. I'm down to nubs. Let's get a good definition of it. This one is almost what your book says. It may be exact or I may have modified it. I have my own preferences. The maintenance of a relatively constant internal environment. And you could add something to this if you wanted, in spite of a changing external environment. Last part's not necessary, but it's illustrated. Maintenance of a relatively constant internal environment. Well, let's define these things. We need to know what an internal environment is. What is the internal environment?
Outside the body.
Yeah, it's inside of our bodies. Is it inside our stomach? Is the stomach lumen? Is the stomach lumen where the food goes? Is that part of your internal environment?
Yes.
Is it part of the external environment?
How do you determine what's internal and external?
Yeah, let's define it clearly. With the internal environment, you have, in order to get there, you have to cross a tissue. You have to actually cross an epithelial layer. The lumen of the stomach, just like the oral cavity, you think of your inside your mouth as part of the internal environment.
No, I don't know.
No, this is part of the external world, right? There's the outside air, and it goes into your oral cavity, through your nasal cavity, down your esophagus, small intestine, large intestine, out the anus. That's all really part of the external environment. Or if you want, you could say it's continuous with the external environment. The internal environment is not continuous with the external environment. It is separated by a membrane. So the integument does most of the separation, but also the oral cavity, right? It's lined with that stratified squamous epithelium, respiratory mucosa, the pseudostratified columnar epithelium. That's a membrane or a boundary, epithelium that separates the internal from the external world. The best way to think of the internal environment is, yeah, it's that part inside your body, but it is really the place where your cells live. We're made up of cells, trillions of cells. Those cells live in an internal environment. They live in an environment that is moist and has lots of nutrients. It's a warm environment. It's the place where your cells live. So when we talk about homeostasis, the maintenance of a relatively constant internal environment, what we're talking about is maintaining a nice place for your body cells to live. The body cells don't live in a nice place. For example, if it's dry, what happens to the cells? They die, right? So our epidermal cells are dry because they live in a, well, not a very nice environment, but they're programmed to dry up, but not very nice. Things like neurons, muscle cells, smooth muscle cells, cardiac muscle cells, connective tissue cells like fibroblasts and fibrocytes, they live in an internal environment, moist, warm, lots of nutrients. The internal environment must be maintained so that that is a nice place for them to live. If it becomes not nice, toxins build up, it's too dry, there aren't the right hormones, wastes build up, there aren't enough nutrients, then the cells die. If your cells die, what happens to the tissue? Dies. Tissue dies, what happens to the organ? Organ's going to die, and then you lose the organ system, and with the exception of the reproductive system, we need all of our organ systems in order to function normally. So, maintaining a nice place for your cells to live so that you can live. Relatively constant, that sort of speaks for itself. Does relatively constant mean unchanging? Or does it mean might change a little bit? Yeah, it is going to change. Relatively constant changes slightly. And I'm going to introduce a term here around a set point, which will mostly be the case around a set point. That is, there'll be some value which is the normal or the ideal, like body temperature, 98.6. That's the set point. Can your body temperature rise? Yeah. Can it go down? Yeah, but it's not going to do it very much. It will fluctuate up and down around that set point, 98.6. So that's what we say, it changes slightly. Some variables change a little more, some change a little less. It just depends on how they're regulated, but relatively constant. That's not bad. Just some values. Here's a table from your text 1.2. Arterial pH, bicarbonate, sodium, calcium, oxygen content, urea, amino acids, proteins, total lipid, and glucose. These are normal constituents of your blood. And you'll notice that they give us a range. So arterial pH is 7.35 to 7.45. Relatively constant. It's going to fluctuate, but it's going to fluctuate due to those narrow ranges. Same with glucose. That's a normal fasting level, 75 to 110. It's going to fluctuate within those limits, and it is going to be different for different people. So these are ranges. Blood glucose isn't held exactly at 100. It does fluctuate. pH isn't held exactly at 7.400000. It fluctuates a little bit. So if we have an internal environment where your cells live and we maintain that environment within pretty narrow ranges, a little bit, but not that much, it tells us something. Let's take temperature, 98.6, that's what you all know as body temperature. What temperature is it outside? How about this morning when you got here? What was it? 32. Right. It's 32 outside. Yet inside your body, your core body temperature should be 98.6. If you were dead, if you were a corpse, and you were outside in 32-degree weather, what would happen to your body temperature? It would start going down, and it'd take a while to get there, but it would start going down. The fact that it doesn't go down, the fact that it's maintained at 98.6 tells us something. It tells we have a regulatory mechanism that is active. It is engaged in making sure that our body temperature stays within that narrow limit. How about blood glucose? Well, we can measure it like after eating, and it will be a certain level, and an hour later it will be a certain level, an hour later it will be a certain level, and it might go down a little bit, but it's going to be maintained at a relatively stable level, at least in between meals. That tells us something. Do cells use glucose? Yeah, so cells are using the glucose, but if our blood glucose levels aren't going down, it means that glucose must be taken from somewhere and put into the blood so that when cells take it out, the value doesn't go down to 0. So it tells us it's being regulated. How are we going to regulate these things? We need a mechanism. There has to be some way that our body can take notice of what our body temperature is and then take actions to keep it at that level. Or blood sugar. Have to have some way our body can monitor the blood sugar. Should be at this level. If it's not this level, take some action to bring it back up to that level. Most body variables are going to be regulated by what are called negative feedback loops. In fact, we can think of a negative feedback loop as basically the primary way to regulate body variables. Whatever the body variable is, any of those up there, if it has a relatively stable value, there's probably a negative feedback system that is in place, actively monitoring and maintaining it. So, all negative feedback loops have 3 basic components. 1, there's a sensor. And this makes sense. A sensor is a cell or tissue or organ that detects the variable. Take body temperature. If you don't have anything that measures your body temperature, how do you know if it gets too high or how do you know if it gets too low? You actually have to have something that actually measures it, that keeps track of it. Same with blood glucose. If you don't have anything that can determine what your blood glucose level is, how can your body know how to dump or when to dump more sugar into the blood or to stop dumping sugar into the blood? You got to have a way to know what the actual value is, and that's called a sensor. Sometimes these are very simple, 1 cell. Sometimes we'll just use a more general term. We'll say, like, we'll name the organ, but always a sensor. Detects the variable. Your book's going to say something like this: detects changes in the variable. Some sensors detect changes. Some constantly monitor the variable. It just differs. So I put that in there. When you read it, you might see detects changes, and it might tell you for a loop. It just means watching the variable. If there is a change, that's actually when something interesting is going to happen. So what's the sensor do? Well, detecting information, and it's going to send a signal or send information to an integration center. There's another term that some textbooks use instead of integration center. They'll use control center. It's the same thing. Control center is a structure that has the ability to process information. So it receives the information about the variable. It processes that information. Process. And it has to make a decision about what to do. All integration centers will have to have some set point. They're responsible for determining what the set point is. So for body temperature, if the set point is set at 98.6, The integration center needs to know that. It needs to know, hey, body temperature should be 98.6. It's going to receive sensor information. Let's say body temperature is, I don't know, 97. And then it compares the two. Geez, our body's 97 degrees. It should be 98.6. Let's do something about that. So determines the set point, determines the course of action. Integration centers are in a position to generate commands. How about generates a command or sends commands to our last component of a negative feedback loop, the effector. Effectors are the structures that can directly affect a variable. You could also say that an effector is able to take an action that alters the variable. What sorts of body structures are effectors? What sorts of body structures have the ability to take actions? Muscles. And that could be cardiac muscle, skeletal muscle, or smooth muscle. What else? Heart, that would be cardiac muscle, good. Glands. Yeah. Muscles and glands. Glands could be exocrine, secreting things on the body surfaces, or they could be endocrine, secreting hormones, but glands. Good. Again and again this quarter, or this semester, what we're going to do is, when we talk about regulation, we'll be identifying sensors. We'll be talking about the integration centers, we'll be talking about the effectors, and we'll be talking about what these things do in order to maintain body variables within narrow limits so that you can maintain homeostasis. Let's go through just a couple quick examples. Ooh, no, that one. Now, I love this one. Sometimes I really wonder about textbooks, but that's fine. We'll do core body temp as an example. This is our variable, and the reason I say core body temperature, it's the temperature that's of the blood that's coming out of your heart, your core body temperature, not your fingertips, like when it's cold outside. That's not what we're worried about. Core body temperature is our variable. We know that it's regulated because it stays pretty constant around 98.6. We need a sensor that detects it. The sensor for core body temperature is your hypothalamus. You have a specialized group of neurons within your hypothalamus. You guys remember that is, right? The diencephalon, right above the pituitary gland, that little area. That monitors the blood's temperature as it passes through the brain. So as it goes through that area, it constantly detects it. Is it too hot? Is it too cold? What's it like? It takes that information and then sends it to a control center or an integration center. The integration center also happens to be the hypothalamus, a different set of neurons within the hypothalamus, but it's still part of the hypothalamus, and it gets a copy of this information. The integration center looks at the information. How hot is your blood? How hot should it be? And then can make a decision. So let's say you're hot. So temperature of your body is what? Let's take you up to 99 degrees Fahrenheit. That's pretty warm. It can make a decision. Well, what do you do when you're hot? Sweat is one thing to do, right? So there's one thing we know you can do in order to cool down. Not that kind of hot. What else do you do? What happens to your skin tone when you heat up? Right, you get red, right? So we call it vasodilation. So there's two effectors that we can employ. One, we got sweat glands, those are effectors, and we have cutaneous capillaries, or cutaneous blood vessels. There's another effector. The hypothalamus is in the position to control each of those types of effectors. So it can send commands to its sweat glands, and then the sweat glands really sweat, and then also to cutaneous caps. And I'm just going to write CUT period caps for capillaries, and they vasodilate. Vasodilation brings that warm blood, more of it, to the surface of your skin, so that when you evaporate sweat, you can lose that heat. What's going to happen to body temperature if you're sweating and vasodilating? Hopefully it brings it down. So if you take these two things, it has a negative impact on body temperature and hopefully brings it down. Let's do the same thing, but let's make it cold. All right? So we've got a sensor detecting your blood, but now it's detecting, hey, it's really cold. It's like the 98.6, let's make it 97. Integration center still has thalamus, gets the information, hey, your blood's cold. Now it has a couple things that it could do. Well, if you're too cold, we'd like to warm up your body or at least prevent heat loss. Are sweat glands going to help? No. So we're not going to do anything with sweat glands. Don't tell them to do anything. How about your cutaneous capillaries? You ever seen someone who's like really, really cold?
Pale.
Yeah, pale. Their lips might even look blue. So that's vasoconstriction. This is how most of us regulate our body temperature, just by dilating and constricting blood vessels. If you're really cold, then you'll start shivering. So we have a different kind of effector. We have skeletal muscles. Shivering generates heat. Vasoconstriction prevents loss, or at least lessens. I should put lessens. Heat loss. You're not losing heat, as much heat, and you're generating more heat. What's going to happen to body temperature at 97? You should then be increasing body temperature or bringing it up. If we looked at this over time, and I know people don't do this, shiver and sweat, unless you're like, what, at the ski lodge, like in the sauna, and then you get really hot, and then you jump in the snow until you start freezing, and then you come back in the sauna. Like, normally our vasoconstriction can handle this. So if we fluctuate the temperature in this room, you guys would vasodilate to lose the heat, vasoconstrict to conserve, vasodilate, vasoconstrict. That's pretty extreme. You really start threatening core body temperature to do that. Is it constant or relatively constant? It's relatively constant. It's not constant. It actually changes. And we know there's a negative feedback loop involved because we see increases and decreases. All right, this word negative, why do they use it? This is my diagram for why they use negative feedback. In a negative feedback loop, if we start off with a set point, if there's an increase in the variable, the negative feedback loop will make it go in the opposite direction. So it will have a negative impact on it. If there's a decrease, the negative feedback loop will have an effect that brings it in the opposite direction. So this is why they use the term negative. But there's other terms that I think are more helpful to remember. How about stable? Stabilizing effect acts to return to normal. Those sorts of concepts or descriptive phrases. Negative feedback systems always work to help bring the variable back to what's normal or back to its set point, at least as good as it can. Concept of antagonistic effectors. Not all negative feedback systems have antagonistic effectors, but many do. So it's worth mentioning it so that when we get to them, it won't be a foreign concept. What does that mean? If someone is an antagonist, are they doing things to help you? No. They are working against you, right? My antagonist is always working against me. Antagonists, I always think of just people who are trying to piss you off. They're trying to make you angry. They're working in opposition to whatever you're trying to accomplish. Antagonistic effectors work in the same way. Antagonistic effectors work in opposition to each other. So in this example that we just gave, we had sweat glands, and we had skeletal muscle. With respect to body temperature, sweat glands help cool you down. Skeletal muscle, when shivering, helps warm you up. Are these antagonistic effectors? With respect to body temperature, one helps cool you off, one helps lower temperature, the other, at least when shivering, helps increase body temperature. So with respect to body temperature, these are antagonistic effectors. They are working in opposition to one another. Sweat and shivering are antagonistic. Let's do another one. When I write sympathetic NS, are you guys happy? Sympathetic nervous system? You remember it at least? Some? What is the other word or common phrase for remembering what the sympathetic nervous system does? General actions. Fight or flight response. Yeah, so good. Sympathetic nervous system, fight or flight response. And how about our variable? How about heart rate? What's the effect of the sympathetic nervous system on heart rate? Increases, right? You get scared, your heart boom, 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 goes up. And then the other nervous system, parasympathetic. Rest and digest. What happens to your heart rate if you're just chilling out? It goes down. So are the sympathetic and parasympathetic nervous systems, at least with respect to heart rate, antagonistic effectors? Yep, they work in opposition to each other. This is a good rule of thumb for these two systems. They will often work in opposition to each other. They're antagonistic effectors. So we'll see this again and again. Let's do one more with endocrine. Blood sugar. What's the hormone that regulates blood glucose? Insulin is one. Yep. And for those of you with diabetes or know people who have diabetes, after they eat food or if they're going to eat a lot of food, they give themselves a shot of insulin if they're insulin dependent, so that they eat the food, the blood sugar goes way up, and then insulin is going to do what to blood sugar? It's going to bring it down. We'll talk more in a few weeks about another hormone, glucagon, which does the opposite. If your blood sugar is low, glucagon will cause the liver to release glucose and bring your blood sugar back up. So one brings it down, the other brings it up. They're working in opposition to each other. They're antagonistic effectors. Good. Make sense? Yeah. Really important concepts. Outrageously important because we see them again and again and again and again and again and again and again. Every organ system we're going to have things like this. And if we can nail down these concepts now in kind of a general way, then you've got this nice structure, hierarchy of basically, or a general example of how things work, and then you can just plug in the different organs and the different variables, and you'll be amazed at how much you know with a very little time. If this is a foreign concept, when we talk about sympathetic, parasympathetic, you're going to be, what? Because your child's trying to deal with, they act in opposition? What are you talking about? Or hormones, they act in opposition? What are you talking about? Regulation? Huh? So, go through the examples in the book. There's a nice one on blood pressure regulation, standing up, lying down in the back part of chapter 1. Read through that as an example as how to test yourself whether you're getting these concepts of feedback, sensors, integration centers, effectors, regulating variables. Questions on negative feedback, antagonistic effectors, sensors, integration centers, effectors. Yeah.
Sorry. So the sensors can be organs and cells and tissues.
Yeah. So for some of our organ systems, we'll just actually name the organ. We won't actually have to make it more detailed. Sometimes we'll talk about a specific cell or a group of cells. It just kind of depends on the level that we work at. But yeah, it will be an organ or part of an organ that is very specialized for detecting the variable, like the hypothalamus, certain group of neurons in it. We won't worry about naming those neurons. We'll just call it the hypothalamus. I think it's in your text. It will have monkey names, which I don't bother to memorize because it doesn't mean anything to me. Maybe in the future it will mean something, and then you can learn them. Until then, just call it hypothalamus. Yeah.
Thank you.
We don't do it in 5 minutes. Not a whole lot. A little bit. Let's say a couple words about variables. Do you guys know what I mean when I say variable? A variable is what?
Anything that changes the body.
Anything that changes, and what really matters here is anything that changes, how about quantitatively, so really, things that you can assign numbers to. Blood concentrations are great, so blood pH, blood glucose levels, we can actually assign a number to them, and then they change. Change, we can then regulate them. If they get too high, we can bring them down. If they get too low, we can bring them back up. The other nice thing about quantitative variables, measurable variables, is that patients who come in, you will take, blood panels on them. You'll be able to measure their values. So if they come in and they've got a series of symptoms, they're peeing a lot, they're really thirsty, you also notice maybe that they're overweight. If you measure their blood glucose levels, you might not be totally shocked if it's highly elevated. And then you can say, well, you probably have type 2 diabetes depending on your age. Probably. You don't know for sure, but you were able to measure a variable. It was out of whack, and you can assume, well, if that variable is elevated, are there negative feedback systems operating normally? No. There's something wrong with that. Not always cut and dry. Maybe it's blood pressure. They come in and you notice that they have high blood pressure. Well, what does that tell you? Well, you know that something's not right, but there's a lot of things that could be wrong. Maybe their kidneys have failed. Maybe their blood vessels are just really, really firm, and they can no longer adequately detect the pressure when it gets too high to bring it back down. So you may not be able to tell exactly what the disease is, but you know that there's something going on, at least with measuring, or say, regulating that particular variable. So that's the other real value of quantitative variables. You can measure them, and if they're off, probably some sort of disease condition or other condition that's going on in the patient that you can hopefully figure out what the underlying cause is and then hopefully treat it. If they got high blood pressure, maybe diuretics, for example. All right. I don't really have time to go over positive feedback in 3 minutes, but I can show you a fun little picture. This from your textbook? Yes. Sorry, I teach at another school, and sometimes I steal from different textbooks. I think this one actually came from you. So this is what we'll talk about next time, positive feedback. Positive feedback loops do exist. They are rare. There are not that many. Many of them don't have all the components that make them nice examples of a feedback mechanism. This is one that does. So I want you to, between now and next Thursday at like noon, look at parturition, childbirth, and try and identify for me what the sensor is, what the integration center is, and what the effector is. Just those 3 elements, and then that's what we'll start next time. So identify the sensor if you can, the integration center if you can, and what the effector is. Got a couple minutes for questions or thoughts if you got them, but that's a great place to call it good for 48 hours or 47 hours. All right. I should also say that if you wanted to take the other instructor, her class has got like 10 students in it. So if you wanted a smaller class, you could switch into her class. And sometimes smaller classes are nicer. Just a thought. I don't want to chase everyone over there either. See you on Wednesday.